Yes, this is the first question, and I want you to try answering it. RNA molecules can be genetic material, messenger, enzyme, all of the above. You can type in your answers, but or say it. Typing is better, I guess. Or one person can say the answer. Anybody?
I'm not sure about the enzyme part, but genetic material messenger. So I'm gonna go for all of the above.
Okay. Maybe I should post the question here, and okay, to give options will be difficult. Okay. Yes, they can act as genetic material, where we have seen that is they can in RNA molecules. Sorry, in viruses, some of the genetic material is RNA, so they can act as genetic material. Their primary function, as we know, mostly is as messenger mRNA. And there is one part that I haven't discussed. RNA molecules can also act as enzymes. Those are referred to as ribozymes. I think it will come up in some other part, so we will discuss at that time. But otherwise, you can also think of it. I mean, just so the answer is all of the above. Okay, RNA molecules can. We have examples of RNA that can act as genetic material, as messenger, as enzymes. Even when we are talking, when we come to the lesson on translation, the ribosome is made up of several rRNAs. And the core of the ribosome is actually made of, I mean, the interacting parts or the ones that seem to mediate the reactions are made up of RNA. So those are referred to as enzymes made up of RNA are called as ribozymes. There are also some people have chemically made something like DNA zymes, and there are some people have made about antibodies that can act as enzymes. They are called as abzymes. These are just um, for your knowledge, just to know those things. Okay. So let's see the next question. Um, this is a little bit of calculation probably, so I would want you to um, these are already known examples. We have already done these, but it will be a good idea if you can try to do these fast. And yes, which of the following is the complementary RNA strand for the sequence, whatever is given? As soon as you know, you can tell the answer, and then we can go ahead.
D.
Yes, please.
Option D.
Option D. Can you explain why?
G is paired with C and A is paired with U because it's RNA sequence.
That's right. I think we already did this question exact same as such. Always, I think wherever possible, you should have your paper pen. I mean, there is something like pinking on paper. If you're that gives you much more flexibility and accuracy actually. So you don't make any mistakes. In CSAR, GATE, and other types of competitive examinations, there is negative marking. That means every time you miss, I think you miss somewhere around 1/4 or 1/3 of the marks that you have gained. Most people miss out because of this negative marking. That negative marking also, I mean, is an indication of your confidence in your answer. Okay, you have to be confident. but you should also be accurate. Yeah, if you are very confident and saying it wrong, then your accuracy is less. So the negative marking is actually testing your confidence levels and your accuracy at the same time, right? Otherwise, if there is no negative marking, you can put any number and just come out, and maybe by fluke somebody might actually qualify. This negative marking plays a big role in that sense. So anything that we are learning should be highly accurate. So instead of memorizing or thinking in the air, it's always better to think on paper if you have it, and so try to write the answer. You can determine the answer. If I put a T here, it is wrong because the question says it's about the complementary RNA strand. Then if you see G, C, U, sorry, A, U, C, G, A, U, C, and it should be in anti-parallel. So this is 3 prime and this would be the 5 prime. All the options are given from 5 prime to 3 prime, right, in this direction. So you should be then able to read in this way, and that is how D is the correct answer. Yes, the answer you said is correct. And then we have also done something like this, which is here. We have also done something like this. So please try to address this question. Anybody?
Option
Anyone?
B is false.
Option B is
Option
false.
B.
Yes,
B,
that's
sir.
correct. Yes, you're right. So you should be, when you're writing CSA or these competitive examinations, you have to read the question properly. And I think you have about 180 or 1.5 hour or so. I think now the rules might have changed. It's supposed to be something like this. You get about 1 minute per question, or maybe 2 minutes, depending on I mean, you have part A, part B, and part C in CSA or UGC net. Part C questions are all research methodology, research based questions. So you require more time, and there are also marks are actually more for them. So read carefully, be aware of what is happening, whether the question says false or so, and read all your options all the time, and then you can narrow it down. Elimination is whichever doesn't fit, you remove them, and the remaining you will put. So I think this is the answer. We have already done this before too. So I'll just add one more. Anyone with this question? DNA pol 1 has 5 prime to 3 prime exonuclease activity. There are 3 things that are given, or 4 things. You have to pick the options. Anybody? What are the functions of DNA polymerase 1? Right? It's not the one that is predominantly the DNA replicating enzyme during replication. We discussed a lot of it. It is a polymerase, and polymerization always happens from 5 prime to 3 prime direction. The reverse of it is never going to happen. Most DNA polymerases also have exonuclease activity, and that exonuclease activity is usually 3 prime to 5 prime 1. That is where the exonuclease activity happens, right? So if there is a wrong base added here, then using the exonuclease activity, that wrong base is removed, and the the remaining, again it resumes polymerization activity. So it is a 5 prime to 3 prime, 3 prime to 5 prime exonuclease activity. Most DNA polymerases have that. I want you to also find out what is meant by Klenov fragment. I'm not going to discuss that now. You should slowly start becoming self-sufficient in acquiring, finding information and trying to do it by yourselves. Okay. The goal of education, I don't think, is to just teach. It is rather training you to teach yourselves. If that is attained, you will forever be self-sufficient. You don't need anybody to teach you something and open it and reveal every detail and all. So I want that you gain those capacities. Keep that in mind, and try to work towards improving your skills. That is learnability. You should be able to learn by yourselves. Well, then we also learned about DNA polymerase 1, that is special. It is also involved in removal of the primers during replication. And for removal of primers, it has to use 5 prime to 3 prime exonuclease activity. Say, for example, this is a primer and RNA primer that is base paired here. And to remove it, it should degrade from 5 prime to 3 prime exonuclease activity like this. So DNA polymerase has polymerization capacity 5 prime to 3 prime, exonuclease activity 3 prime to 5 prime, which most polymerases. And something special in DNA polymerase 1 is it has 5 prime to 3 prime exonuclease activity as well. So what is not possible is this one is not possible. There is nothing called as 3 prime to 5 prime polymerization activity. It simply doesn't exist that way, at least not in natural settings. Maybe in lab you might be able to do those by using in chemistry probably, but otherwise in biological system, polymerization is always from 5 prime to 3 prime. So the answer then would be where is it? Which one? Yeah, C would be the answer for it. Okay, you have any questions on that? Okay. So try to answer this question. If you don't know, it's all right. Maybe we didn't discuss enough of it. But I still want to, because the information is given to you. Sorry. So the molecular weight of a protein is 3.3 kilodaltons. What is the length of the gene in base pairs? All right, I'm giving you a molecular weight of a protein. This is a protein. I'm giving you the weight of it, and I'm asking you to find the length of the DNA in base pairs. I didn't ask you in micrometers. That would be a little more, a higher level added to that. So you should always remember from your biochemistry, you may have heard that the average molecular weight of an amino acid is 110 daltons. Okay, that is for calculation purposes. It is always used, average molecular weight of amino acid is 110 daltons. Remember that. So if I know the number of, we have some things to discuss, but still I put up this question because you already have learned it some in biology for engineers. Even if you don't remember, that's all right. This is just a trial. So as we progress, you will have a better understanding at that time if you didn't understand now. So first of all, what is the number of amino acids in this protein? If I want to do that, I should take the total of 3.3 kilodaltons. So I will just take it as daltons. So you should always remember that. Sorry, I made a mistake. So basically, if you want to do it, I'll put it like this: 3.3 into 1000. And then that is because I'm now converting kilodaltons to daltons. Always keep track of the units, because otherwise it's likely that we might make a mistake. Number of amino acids is 3.3 into 1000 daltons by 110. Okay, if you have a calculator, you can quickly do those things. Okay, if it is for simplicity, maybe we'll do it this way, right? And that will be, how much will that be? 3.3 into 100 by 11. That's the answer. So the total number of amino acids is 30 amino acids. That's right, 30 amino acids. And if you know central dogma of in molecular biology, you know that 3 bases code for. If I know the number of amino acids, I should be able to know how many codons are there. How many codons? Each codon will be, each amino acid is coded by 1 codon, right? So it will be 30 codons. And each codon is about 3 bases. So 3 bases into 30, that will be about 90 base pairs, right? I mean, basically you're talking about, when you calculated, you're calculating one ORF here, start codon to stop codon, right? Whether it is in bases or base pairs is much better to say. So the answer then would be 90 base pairs. That is the answer for it. Okay, I'll quickly tell you how it is done. You have to calculate the number of amino acids. Number of amino acids is the first step, and that you use the 110 kilodaltons. 110 daltons. Average molecular weight of amino acid is 110 daltons. From there, you should be able to. You should be able to calculate the number of codons, and from there you will get the base pairs. Right? And again, if I have to ask you, tell me the size of the gene in micrometers, then you would have had to have done one more step. Okay. Most of the GATE life sciences or biotechnology questions will be like this. They do not directly ask you any straightforward questions. You will have to employ certain level of intelligence there. You don't have to memorize everything. You should be able to determine from there whatever information is given. From that, you should be able to determine the answer. Okay, let's move on to the next one. A double-strand DNA was found to contain 30% of G. What is the percentage of T? That's a simple question. Please answer faster.
20%.
Yes, if G is 30%, C is also 30%. Then the remaining is 40%. So A plus T would have been 40%. So the question for T alone, it would be 20%. That would be the answer. Okay. Let's see the next question. It is a little bit of memory based. I hope you will be able to make it up. Structures of nitrogen bases. Deamination of thymine results in uracil. Demethylation of thymine results in uracil. Deamination of methyl cytosine results in uracil. Demethylation of thymine results in cytosine. Each of the options is like one question, right? And we should be able to figure out which is right and which is wrong in that. What shall we do? You can Google it up if you want. I want you to find out what is the answer for this question. I will wait for 5 minutes because I thought I made you practice enough of these structures. So, what is the base structure that one has to remember? It is uracil. And you have O and O. I'm not solving the double strands. So this is uracil, and the numbering is something we need to also remember. It would be 1, 2, 3, 4, 5, and 6. If I add what is thymine? How to remember it? 5-methyl uracil is thymine, right? If I were to add a methyl group here, that would have been thymine. Okay, so demethylation of thymine, that's the answer. Demethylation of thymine results in uracil. If I were to remove the thymine from here, then that would have resulted in uracil. Okay. And at one point, we will also get methyl cytosine. And what would happen if deamination of methyl cytosine occurs? We will learn that in a later class. Yeah, this is something, the question that we are going to do now is directly relevant to the question that we have done, the lesson that we have learned in the last class. So I want you to answer this question fast. The DNA content of a gamete after meiosis is 8 into 10 to the power 6 base pairs. What would have been the DNA content of the parent cell in the G2 phase of the cell cycle? I won't answer. I'm waiting for you to answer. Anybody? It's a simple question, actually. Sorry, anybody? Is anyone answering?
32 into 10 power 6 base pairs.
Okay. Why do you say so?
2 into 2n, because after meiosis we have haploid,
That's right.
and after G2 phase we have 2 into 2n.
Yes, yes,
So,
keep saying. You're right, keep saying.
so 8 fours are 32.
You can think of it. Yeah, your answer is correct. Okay. Say this is during, after meiosis, you have haploid cells. And the question says there is about 8 into 10 to the power 6 base pairs. I'm not writing the whole. So that means the cell that was undergoing in G2 phase should have had, this is right, 32 into 10 to the power 6 base pairs. Okay. That would have been in G1 phase. What would it have been? It would be 16 into 10 to the power 6. That is 2n in G1 phase, kind of 4n, not exactly 4. You shouldn't say tetraploid. Okay, don't call it tetraploid. It is not tetraploid, but for the sake of understanding the genome sets, you can write 4n. And here you have 4 cells, each of them is haploid with N, right? So the answer is 32 into 10 to the power 6 base pairs. Okay, the next question is something interesting. You can remember we must have done this before, and please solve this.
One 30 hydrogen bonds.
Okay, how did you solve that?
We have a 30% of G in 100 base pair. So 30 will be guanine and 30 C. So between each G and C, we have 3 hydrogen bonds. So 30 into 3, it's 90.
Um, G is 30%, right?
Yeah,
So which would say, sorry, this is C will be 30. 30% of when you say 100 per 100, how would you say that? One second. Uh, Sorry. Yeah, continue. 100 base pairs. 100 base pairs, and 30% of it, I will just say there are 30 Gs. Is that clear? Is that simple? Which
in
would
30
also,
C,
yes, and you'll have these are 30, and the remaining is
20A and 20T.
20A, which is 2 hydrogen bonds, and 20, right? So if I know 30 into 3 equal to 90 hydrogen bonds in between these two, G and C. Between A and T, I have 20 into 2. That's equal to 40. So 90 plus 40, that's 130. Yes, 130 should be the answer for it. Okay, so let's get going. This is something, again, interesting question. If you can think of it, use any resources right now if you want to, but figure out the correct answer. The difference between a single stranded DNA and a single stranded RNA molecule with the same number of bases and same base composition is DNA has more number of oxygen atoms than RNA. RNA has more number of hydrogen atoms than DNA. DNA has more number of carbon atoms than RNA, and RNA has more number of phosphorus atoms than DNA. What is the correct answer for this? I think for this question, if you're already thinking, that's good. The answer is first we should try to recollect the differences between DNA and RNA. DNA has thymine, right? DNA has thymine, and I'll just put capital T. RNA has uracil. And what is the difference between them? It is CH3, a methyl group. And we have one more problem with ribose and deoxyribose. In DNA, it is deoxyribose. and RNA has a ribose. The difference is one oxygen atom, right? So now try to, that is what you should always remember. Instead of remembering as thymine, the DNA has T and RNA has U, you just, it's one additional more. That DNA, the difference is one methyl group between thymine and uracil. And deoxyribose and ribose, it's one oxygen atom. So probably there are more than one option that could be right. So think of it and then you can answer it. DNA has more number of oxygen atoms. That is wrong, right? DNA actually has less number, considering this. RNA has more number of hydrogen atoms than DNA. Uh, no, right?
No, sir.
Yes. Yeah. So that is also a wrong option. DNA has more number of carbon atoms than RNA. It seems like
It's
that.
right.
Yes, this is there. So that is correct. RNA has more number of, that is wrong. We don't have to read about phosphorates. It's always the same. Okay. So your basic knowledge, when you are acquiring the basic knowledge, pay little more attention, and then many things will become much easier. And actually, it will be a lot of fun trying to understand these questions. Uh, okay, let me try this question. Just give me a minute. This should be a lot of fun, actually. Solving questions is, you have to take every exam as if, I mean, especially those that have multiple choice questions. It's a lot of fun. It should be a game between us. Denaturation of double-stranded DNA is favored by low pH, high pH. If I haven't discussed this, this is a good time to know about it. Anybody?
High pH.
Why is that?
Because the DNA is, more stable in a lesser pH.
Why? Why is it stable in less pH?
Because of the acidity. It's a negative molecule. The
Yes. So
So in usually negative molecules are more stable in a lesser pH because they tend to exchange the protons.
yes.
So
So
when you
when
give it in a higher pH, it
uh,
favors denaturation, as in
yes, so where do you think hydrogen bonds will be more stable? I mean, I don't need to necessarily think about strict chemical ways. You can also think of it in simpler terms too. What you said is, I would agree with that. And always try to remember how we feel about, or how we rational put rations to it, right? How we rationalize an answer. So I'm trying to draw the bases here and base stacking and so on. It's not that great, but yes. So here you may have these hydrogen bonds, hydrogen bond donors, and some acceptors will be there, right? If I had more number of um, OH, say when you say what is pH, it's negative logarithm of hydrogen ion concentration. So that means when I say high pH, that means the hydrogen ion concentrations are actually very less, which would relative to, or which would mean that you have more of OH, right? In those conditions, you may have more acceptors of the hydrogen bond. And in that case, it's not necessarily this molecule base that is competing with for the hydrogen bond. You can also have OH that is competing for these hydrogen bonds. That is how I tend to believe. I tend to remember stuff, right? When there is basic um, or alkaline conditions, then the DNA tends to denature more. compared to that of other pH. Okay. So when we do in the lab, we have an experiment of isolation of plasmid from bacteria. In that particular one, we will discuss a method called as alkaline lysis method. Right? We will lyse the cells in alkaline conditions, which is also the case where the DNA gets denatured. Okay. So the answer for this should be this one. So here is one another question I want you to answer. Try to recollect whatever you learned before in other courses too. Regarding the reading of genetic information, various processes of central dogma of molecular biology, pick the false statement. DNA polymerize DNTPs only in 3' to 5' direction. RNA polymerizes NTPs only in 3' to 5' direction. Ribosomes polymerize amino acids reading the mRNA in 3' to 5' direction. None of the above. That's a simple question, I think. Anybody? What's the answer, please? Somebody? No polymerization happens from 3' to 5' direction, right? Wrong, wrong, and wrong. Everything is always happening or being read 5' to 3', right? So none of the above is the answer for it. Try to solve this. This is directly about nucleosomes, the lesson that we have learned in the, we finished in last week. The composition of a nucleosome is Maria, please. That's a simple question. What is the composition?
8 subunits, sir.
That's right. One nucleosome has 8 subunits, 2 of each H2A, H2B, H3, and H4. There is a certain, again, you have to remember us, the most appropriate way is to put into 2 into H2A, H2B dimer. There are 2 dimers and 1 into H3, H4 tetramer. Right? Every nucleosome has those things. So you have total of a 4 here, and total of 8 subunits. Yes. This is a simple question. Try to solve. This is a knowledge-based question rather than intelligence in a way. The protein that increases the processivity of DNA polymerase during replication is? Anybody? Yes? So the protein, it is sliding clamp. What is sliding clamp? It is the protein that encircles the DNA like this, to which the DNA polymerase is bound, and then it keeps polymerizing. Okay? So sliding clamp is wound around DNA, and DNA polymerase is interacting with sliding clamp, and then that increases the processivity. I hope you remember what is meant by processivity. Processivity is the phenomenon where it's about how many number of subunits are added before DNA polymerase dissociates from the DNA. Something like DNA polymerase 3 has high processivity, and DNA polymerase 1 has very low processivity. So it's mostly about being in contact, and sliding clamp is actually helping uh, increase the processivity. In we also remember PCNA. In eukaryote, sliding clamp is called as PCNA, proliferating cell nuclear antigen. Okay. I'll give this one question which we haven't discussed, but I think some of you could answer it. It's not a multiple choice question. Try to answer the question here. Anybody?
Yes, sir, it is possible.
Okay, how is that?
Uh, for A, if they have alkyls as IA and I small I, that is A being dominant and small I being recessive, and similarly for B blood group also we have B being the dominant and small I being the recessive. Two recessive combined together can form blood group O.
That's right. You're right. So I'll just for ease of understanding, I'll put A. If there are in genotypes, we will talk about it when we are discussing about Mendel's laws. Um, we are all diploid, so we have two copies for everything. So A, and if one gene is encoding for A blood type, and the other one is not coding for anything, something like O, right? What is the phenotype? This is the genotype. What are the types of genotype? Okay, and phenotype is about character. What type of character or trait is being exhibited? Genotype is what type of genes are present. So we have, if the person has A and O, the phenotype would be blood group A. If it is B and O, the phenotype would be B. Why is that? B is dominant over over O. A is dominant over O. For simplicity, I'm just saying that way. If a person has O and O, then that person's blood group would be O. If a person has A and A, then blood group is A. Phenotype is A. Like that, you can have any of these combinations. You will also have A and B, and that person will have AB blood group. Okay, so one is dominant, one is recessive. Here you'll say this is dominant. A is dominant. In this case, B is dominant. Here O is recessive. Okay, and A, these two, when both the alleles are A type, say for example the same similar copies, then you call them as homozygous. This is a good way of trying to introduce some of the terms. So when the class comes, we will be able to do that. So here is O and O. So this is also homozygous. Here you have A and B. A is dominant and also B is dominant, because that is where you see in the blood group. So there those two are co-dominant. right? If they both are expressed, then you call it as codominant. And you have one A and one B, so it is heterozygous. Similarly, here you have A and O, B and O. Those two are also heterozygous. Okay. So I'll just erase this. During gametogenesis, what may happen is if the person is if they are like this, so there are several possibilities that may happen between a male and female like this. The child may get one A and one B from the other one, or a child may have A and O. So the blood group would be, here the blood group would be AB, here the blood group would be A, and if there is another chance is B and O, the blood group would be B because B is dominant, and there is another chance that is O and O, where the blood group would be O. So if the parents were heterozygous for A and B like this, then there is a possibility that the child could be of O blood group. Okay. There is, I think this will be the last question. I thought I will be able to cover more, but that's all right. This will be the last question. I want an answer quickly. Genome of a virus was found to contain 30% of A, 50% of G, 10% of C, and 10% of U. Pick the most appropriate conclusion about the viral genome.
It is single stranded RNA.
Why is that?
Being a single strand, it can have various composition of bases.
That's right. What are those rules? What is the term used for
Chargaff.
base composition? Yes, Chargaff's rule, right? What is it? It should be G is equal to C and A equal to T, right? They're always that way. And this is applicable only to double-stranded DNA or double-stranded RNA. Okay? And your answer is correct, of course, Mazuk. Here, it is, we are saying A is 30%. Where is it? Sorry. A is 30%, G is 50, C is 10, and U is 10. That means it is not obeying Chargaff's rules. That means it must be either, it must be single-stranded, right? And you're saying there is uracil, which would mean that it is RNA and not DNA. So C is the correct answer. So if one wants to master any of these competitive examinations, I don't know, I hope you know that our seniors, current fourth years, there are 6 or people who have qualified GATE, and some of them got very impressive ranks, 20 and so on, all over India, and that is in biomedical engineering. So you will be eligible, or the best one shot you have is at biomedical engineering. But in the past, most of our people have taken life sciences. If your chemistry is good, yet +2 chemistry is good, that should be sufficient for writing GATE life sciences. You're also eligible to write biotechnology GATE. There is another ecology and evolution. There is one more. You can, I think you can take multiple, more than 2 or more than 1 stream. So the competition is less, I think, all over India, because it's a new one. It is less. This is one heavy competition. These two have very high competition, but still we are very good. At one point, one of the biotechnology student was all India rank 1 topper. That was in 2013 or 14. I can't recollect it. I want you to be open to those and never let your enthusiasm for competing with other people, about the rest of India. Your classmates are not your competition. It's not about other deemed universities. It's about all India level. Where do you stand? So please keep in mind that you have to nurture yourselves with a mindset for competition, so that you will be able to compete and better yourselves. And my happiness really with about students' progress is about how many competitive exams did they qualify. If you give me 1 minute, I will just tell you one thing and I will let you go off. So there are two brothers. Okay, the one has qualified, one was doing PhD in India and another was doing PhD abroad. Not that anybody is big or small, and this person happens to be, say, person X. Person X happens to be a friend of my friend. So my friend asked him, why didn't you go abroad? You seem to be, you are better than your brother Y. And the answer of X is something like this. I qualified CSIR exam. I got admission in India, so I joined. My brother tried hard to qualify CSIR. They didn't get. That's why he's going abroad for PhD. Nothing less of, I'm not saying anything less of going abroad. What I'm talking is highlighting the competition that is there in India. About 75,000 people write the exam. Around 1,000 or 15,000 people are given, I think, sorry, around 1,000 people are given JRF. That is, they will pay you salary about 33,000 per month for 5 years. I don't need you to even take up this job. If you want to go abroad, go abroad. But write the exam, qualify it, and show that you are capable of better than anybody else. It comes to about 0.1%, I think. That if you qualified CSAR, or say GATE even, then you are about 1.1% of all the intellectuals that attempted that exam, and you are in the top cream layer. That says something. That is competition. You can write GRE, TOEFL, and all. There is no competition there. You get what you get, and nobody is can steal your place. But here, you're all competing for just these 1,000 JRFs, and there will be so many people trying to do that. In the past, our bioengineering students, many of them have qualified CSAR, and they are now pursuing PhDs. Some of them qualified but didn't take up this. They went abroad. That is also fine. Just keep yourselves open about it and actually enthusiastic about it, and continuously you should keep it. No one should drag you down from the competing spirit. You have to nurture it yourselves, and you're more than welcome to meet any of your faculty members or me or anybody. Try to seek advice, talk to other people about it, and listen to only encouraging and motivating suggestions. Don't take anybody who says you can't do it. Don't ever listen to them. At the most, we might not qualify, nothing to lose. At least we had the guts to face the exam. Okay, we'll discuss in the next class. We'll talk about Mendel's laws. Take care and have fun.
Thank you.
Yeah, bye-bye. Bye.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Have fun.
Thank you, sir.